Hi everyone, and welcome to today's lecture where we're going to focus on the control of carbohydrate metabolism and the citric acid cycle. So the first thing to know when we head toward this information is to look at the molecule called glycogen. Now, glycogen is basically, you can think of it as the stored form of glucose. So as you can see here and in this figure here, it's basically a polymer, made up of a bunch of monomers, or you can say a polysaccharide since it's a carbohydrate. So poly meaning many, saccharides linked together, and you can see that here and each of the little circles here representing all of those individual monosaccharides linked together. And so it's basically this big branched collection of monomers that makes up this big polysaccharide. Now, you can kind of think of it as similar to starches that are in plants. And the main idea when it comes to glycogen is to remember that it's especially found in the liver and in the muscle. And this is important because those are two areas where you're going to see that it's important to be able to degrade or break down this glycogen structure to obtain glucose or to ultimately get energy. And so when I say how does glycogen structure impact being able to break it down and obtain glucose or eventually energy from it, when you see this branched structure, you can see that basically you're able to get several glucose residues or glucose molecules from this one big branched glycogen. Okay, so it becomes very valuable rather than just having a single line of glucose linked together and only being able to get a limited amount at a time. Because if you think linear, you'd only be able to break off one from each end at a time, whereas branched, look, all these red circles show that at one moment you can break off all of those glucose pieces because of the branched nature, rather than if it was linear and you'd only be able to get one end, the other end at a time, so two at a time. Here's a recap of the information, some of the information that I mentioned, and some of the answers from the previous slide. So now, I kept mentioning how glycogen is basically stored glucose and stored energy that can then be broken down. It's important to note that rather than just instantly obtaining glucose, when glycogen is first broken down, it's broken down into glucose 6-phosphate. And the breakdown of glycogen basically differs depending on where it's getting broken down from. So like I mentioned in the last slide, glycogen is primarily found in the liver and in the muscles to be broken down. And so in the liver, when glycogen's broken down, it's hydrolyzed to, it's basically broken down to glucose 6-phosphate, which is then hydrolyzed to give glucose directly to pump that into the blood and to restore the blood sugar, your blood sugar level. Whereas in muscle, instead of hydrolyzing that glucose 6-phosphate to give glucose, instead the glucose 6-phosphate goes into glycolysis, and glycolysis is the pathway that will basically allow for a lot of energy to be created. Okay, and that's important because your muscles, when they're contracting and when you're moving those muscles around, they need energy. So going into glycolysis is very valuable for the muscles. Now, when we talk about the metabolism of glycogen, or basically the conversion of glycogen, there are three reactions that I want you to be aware of. The first one is demonstrated in this figure over here. And basically what happens is a glucose residue is broken off from the glycogen, and it reacts with a phosphate ion. And when they react, that gives you glucose 1-phosphate. Now this is called sorry, I mess this word up all the time. You call it phosphorylasis or phosphorylasis. And ultimately it's catalyzed by this enzyme here. So glycogen phosphorylase, because again, it's a phosphorylasis reaction. And when you do this, you end up, like I said, with glucose 1-phosphate. But now you're going to see, even in the previous slide I had mentioned, the more important molecule is glucose 6-phosphate. So this guy needs to be converted to this guy here. And that's the second reaction. The second reaction is basically an isomerization reaction where glucose 1-phosphate is converted to glucose 6-phosphate. And isomerization, or the isomerizing of this guy to this guy, that basically means that both of these have the same chemical formula. They just have a slightly different arrangement of excuse me, of where you will find certain parts. So in this case, the phosphate ion that we saw from the previous area. And so this is the second reaction is catalyzed by this enzyme here, phosphogluconeogenesis. I mess up on that enzyme too. Now, when we saw glycogen in the previous slide, one of the things you noticed and that was important with its structure, was the idea that it was branched, and we emphasized why that's significant. So now, if you're converting or metabolizing glycogen, the last thing that you're going to need to do is to make sure that you debranch that structure. Okay, so to completely break down glycogen, you have to debranch the structure, and that's shown in this figure here. Basically, when you think of the branching of glycogen, you have the hydrolyzing of the glycosidic bonds, and this occurs at those glucose residues at the branch points. And basically what that means is, when you picture glycogen, it's like a whole bunch of glucose residues stuck together. And so you need to have a breaking of those bonds. And so there's something called glycogen phosphorylase that will cleave certain linkages in the glycogen to debranch and to remove the linked branches. And I'm trying to see if they had wrote the enzyme on here there, so it just shows the phosphorylase action there. Okay, so please be aware that the conversion of glycogen requires 3 reactions, and emphasizing that that includes the debranching, and it includes being able to isomerize glucose 1-phosphate to end up with the very significant glucose 6-phosphate. Now, with everything in science, whenever you have one reaction and one outcome, you have to have some way to counter that. And so, since we have glycogen conversion, or metabolism, which is basically the breakdown of glycogen that we talked about in the previous slide, you also have to have glycogen synthesis, so glycogen formation. So now, what's interesting about this is it's not actually just the reverse of the breakdown of glycogen to glucose. There are some differences and some different stages. So when we think of the glycogen synthesis, the first thing I want to emphasize is that in this process, in the beginning, it requires energy. And that energy is obtained by hydrolysis of UTP. What UTP is, or what that stands for, is uridine triphosphate. Okay, so it's a nucleoside triphosphate. And In the first phase or the first step of glycogen formation, what you have happening is you have glucose 1 phosphate, which we've mentioned before in the previous slides, that will react with the UTP to form what's called uridine diphosphate glucose. Okay, uridine diphosphate glucose, and that's what's shown down here, and you'll see it written out in textbooks as UDPG. And what happens is that UDPG, which I have highlighted here, ends up attached to a growing chain. So you see that in this formation here. So you have UDPG over here getting linked onto the growing chain of glycogen. And then in the end, because you saw in the previous slides that a big aspect of glycogen structure and significance is its branching formation, finally the big chains that have been formed have to be put into branch form, and that's accomplished by the branching enzyme. Now, I just emphasize that when you have one process in biology, you have to have something to counter it. So we talked about first glycogen breakdown, and then we talked about glycogen synthesis, and they accomplish the opposite task of each other. And so when you think of glycogen metabolism and control, it's important to prevent having both of these opposite reactions occurring at the same time. You know, you have to go based on what a cell or what that tissue or muscle or liver needs at that moment. Okay, so you don't want synthesis and breakdown happening at the same time. And when it comes to preventing simultaneous glycogen synthesis and formation and breakdown at the same time, the big controlling factor is the control of the enzyme glycogen phosphorylase. And glycogen phosphorylase, that's the enzyme responsible for glycogen breakdown. And it's able to be controlled allosterically, which we've talked about in the enzyme chapter. That's, allosteric control is when you get like a change of shape of the active site or of the enzyme. And it's also able to be controlled by covalent modifications, which we also saw in the enzyme chapter. If you remember, poisons can do that. Covalent modifications are irreversible, like we saw in the examples of penicillin. And when you think of this phosphorylase and this ultimate control of glycogen metabolism, I want you to kind of use logic to think about what may be inhibiting breakdown, what around the environment may inhibit synthesis instead. And so I highlighted here this condition that is, the ultimate inhibitory condition for glycogen breakdown. So if there's low AMP, high G6P, which is the glucose 6-phosphate, and high ATP, that's going to inhibit glycogen breakdown. So when I say use, logic to kind of help yourself remember this, the trick is think of, okay, glycogen being broken down. Why does a cell want to break down glycogen? Well, in glycolysis, we break down glycogen to get energy, right? You could get 2 ATP from glycolysis, and then the pyruvate or the products of glycolysis can go towards cellular respiration, and you get a whole lot more ATP down that pathway, right? So a cell's going to want to break down glycogen if it needs energy. So if instead there's high energy present, you're not going to want to waste your time breaking down glycogen because you don't need more energy. You have a whole lot of high ATP. You have energy. And whenever you have high ATP, that means low AMP, right? So that's why this is in the condition as well. The other part of this condition you notice is high G6P. Well, we saw in the first step of the breakdown of glycogen, we saw G6P getting produced. Glucose 6-phosphate is one of the intermediate products in glycogen breakdown. And so we talked about previously negative feedback. If you have high glucose 6-phosphate and high ATP, which are both products in the glycogen breakdown pathway, they will then inhibit that reaction because you don't need more of them once you have a lot of those products. It would be wasteful. And then the trick to then figuring out what inhibits synthesis will just reverse each of these conditions here. If these inhibit breakdown, then the opposite will be inhibitory towards synthesis. So synthesis would be inhibited by high AMP, low glucose 6-phosphate, and low ATP, right? Because if you have low ATP, you're going to need more energy. You're going to need to make more energy, more of the, more of the ATP. And so make sure you write down that synthesis is inhibited by high AMP, low G6P, and low ATP. Because at that point, if you have low ATP, you're not going to want to make more glycogen. You're going to want to start breaking it down to get energy, right? Because by that point, you're low on the products that you would want. So then you're going to want to break down and not synthesize. Okay? So kind of use that logic of thinking first, well, what is glycogen for? Well, it could be broken down for energy. When would you need to break it down for energy? If you don't have a lot of energy there. Okay? So now I want to focus on the process of gluconeogenesis for a little bit. And whenever you see genesis in any terminology in the English language, that means creation of something. And so here in gluconeogenesis, you're creating glucose from pyruvate or lactate. Now that should sound familiar to you, these terms, pyruvate, lactate, and glucose, because you just saw all of them being part of glycolysis. But if you look at this figure here, the ultimate goal of these two reactions, glycolysis versus gluconeogenesis, are completely opposite. Okay, so in glycolysis, you're breaking down that glucose and heading toward products of pyruvate and lactate, whereas in gluconeogenesis, you're then using those products to head toward glucose. So you can think of gluconeogenesis as the reverse or the opposite of glycolysis, but just remember that the reactions themselves are not completely the reverse of each other. Okay, there are 3 steps highlighted in this figure. This box here, this rectangle here, and this rectangle here, where the reactions of glycolysis cannot just simply be reversed. Okay, so even though the two processes have the opposite goals of each other and can be looked at as the reverse of each other, just remember that chemically speaking, the reactions themselves are not 100% reverse of each other. There are 3 steps that are not true, just, hey, let's just reverse this. Okay, now that you're familiar with glycolysis and the term gluconeogenesis, I want to give you a body connection, big picture idea. I want you to circle, star, highlight this term here. Okay, make sure you write in your notes the Cori cycle. Cori is named after the guy who actually found this cycle to be. So now what the Cori cycle is it's shown here in this little diagram. It's also shown in this one too, but this one I think helps you kind of visualize it nice and quickly. The Cori cycle is basically the idea that you have this cycling between the liver and the muscles. And what happens is the muscles produce lactate, okay, during glycolysis. And you always hear that about, oh, you got to be careful when you're working out, the lactic acid, build up and all that. So picture the muscles using glycolysis and forming lactate. And then in the Cori cycle, that lactate, if you follow it, then goes to the liver, okay? So it's transported by the blood to the liver, and there it'll undergo gluconeogenesis. And so in the liver, gluconeogenesis is going to convert that lactate back to glucose, which can then be carried back to the muscles through the blood, and then can be used again in terms of glycolysis and producing that energy. And then the lactate can go back to the liver, and you get this cycling. And during this process, if need be, glucose can be stored as glycogen and then can be degraded by glycogenolysis to then, have that glucose to continue to be cycled throughout this. Okay, so please make sure you're familiar with the term the Cori cycle, and that it is the cycling between liver and muscles, where the muscles over here will break down glucose to lactate in glycolysis. The lactate will then go to the liver to be converted back to glucose, where it could then keep cycling between these two. And you notice this allows the muscles to get energy that they need. Okay? Now, before we can look further at the steps of gluconeogenesis, I want to show you what has to happen first. So pyruvate going toward this gluconeogenesis, when we look at this, you'll notice that ultimately we need oxaloacetate to go into gluconeogenesis over here. But the problem is that the enzyme that converts pyruvate to oxaloacetate, which is shown in this figure here, that conversion, pyruvate carboxylase, that enzyme is found inside mitochondria. But as you see, where you want gluconeogenesis to occur is out in the cytosol, okay? And so you first have pyruvate out here in the cytosol. You ultimately want the reaction out in the cytosol, but the enzyme needed for that first conversion is in the mitochondria. Now, the problem with this is that once that enzyme converts pyruvate to oxaloacetate, which we want for gluconeogenesis to then occur, the problem with that is this guy here, oxaloacetate, cannot cross membranes. Okay, it cannot cross the mitochondrial membrane. So how do we then get this guy where we need it in the cytosol for gluconeogenesis to occur? Well, what has to happen is it has to be reduced to what's called malate first. And malate can cross the mitochondrial membrane to then get outside to the cytosol. You can then have the malate converted back to oxaloacetate, so oxidized since it was reduced in here. And then now you can have gluconeogenesis occur. Okay? So I want you to really emphasize in your notes this part, the idea of, well, what do we need to happen? We need pyruvate to become oxaloacetate for gluconeogenesis, but what's the problem? This conversion, the enzyme is inside the mitochondria, and oxaloacetate, once it's in the mitochondria, once it's been produced, it cannot cross the membrane. So please remember that malate is an intermediate that allows for crossing the mitochondrial membrane so that we could then have the oxaloacetate outside in the cytosol where we need it. Okay, so Now that we've seen that conversion to get us to the oxaloacetate in the right location outside of the mitochondria in the cytosol, now we can take a look back at what's actually occurring in gluconeogenesis. So remember, in this figure, if you go downward, then you're looking at glycolysis, and if you go upward, you're looking at gluconeogenesis, because gluconeogenesis is going from lactate or pyruvate all the way to glucose. Now, when we are looking at this reaction, we're starting over here where we just had our oxaloacetate created, right? And so now what you see, the next step to be done is you need PEP created. Okay, that's phosphoenol pyruvate. You don't have to memorize the full name, just know PEP, okay? That is accomplished by PEP carboxyl kinase, which you see over here. And what you'll notice is whenever you see any of the reactions in gluconeogenesis in a beige, yellowish box or rectangle, this represents that this first section over here is one of the areas where it is not just the reverse of glycolysis, because some of the reactions in glycolysis are not energetically reversible. So they're what we call energetically irreversible. And so instead, basically the gluconeogenesis process has to use extra enzymes to kind of get where it needs to go to ultimately be, as if it's the reverse of glycolysis. So we had oxaloacetate that we got, through that malate intermediate. Now we have it converted to PEP through PEP carboxylase. Okay, that's what needs to be synthesized. I'm going to have a recap of the answers of this stuff on the next slide, so don't worry about jotting it down. All right, then after PEP, you can basically go through and proceed as if it's just, the reverse of each of the reactions over here from glycolysis. And then you hit this area over here. And now you've hit the last of the 3 steps that are not the reverse of glycolysis. Okay, so this is one of the 3 steps that's not the reverse of glycolysis. Then you go through the reverse steps, and then you hit the last 2 steps here of gluconeogenesis, or not just the reverse of glycolysis. And what you can see happening in these steps here, this first one, you have the hydrolysis of fructose 1,6-bisphosphate to produce fructose 6-phosphate and a phosphate ion. And that's accomplished by this enzyme over here, fructose 1,6-bisphosphatase. And then the last of the steps, which again is one of the 3 steps that's not just the reverse of glycolysis, this last step is where you have the hydrolysis of glucose 6-phosphate to glucose and a phosphate ion, and that's accomplished by glucose 6-phosphatase. Okay, so circle star highlight the 3 irreversible, the 3 steps of gluconeogenesis that are not the reverse of glycolysis. So that's this one over here, I mispronounced that word so many times, oxaloacetate to PEP through PEP carboxylase. You have fructose 1,6-bisphosphate to fructose 6-phosphate over here. And then the final step, glucose 6-phosphate to glucose. Okay? And I'll write it out on the next slide that the reason why I keep emphasizing these 3 steps is because in glycolysis, when we were at those areas, such as, the first step of glycolysis, which would be up here, the enzymes used in those steps are energetically irreversible. So for instance, what did we say glycolysis, the first enzyme is? Hexokinase. And I said to remember that hexokinase, and I said to remember the last enzyme, which is pyruvate kinase in glycolysis. So those are 2 of the 3 energetically irreversible steps of glycolysis. So I'll have those written out on the next slide so you can just pause and jot down those notes. So as promised, here's that recap. Like I said, these are the three enzymes, the three steps in glycolysis that are energetically irreversible, which is why we had to emphasize those three beige or yellowish squares and rectangles on the figure, because that shows how gluconeogenesis overcomes the irreversible aspect of those three glycolysis steps. So now we're just going to look at a summary of some of the regulators, the control in glycolysis and gluconeogenesis, because you always need things that can help activate and start up a reaction when you need it, and you need things that can inhibit or stop that reaction when you don't need it, and it'll just be, a waste of time and energy and resources. So the first one I want to point out, if we're going downward, we're looking at glycolysis. If we're going upward, we're looking at gluconeogenesis. Okay, so in this slide and the next one, anything you see on the left is a regulator of glycolysis. Anything on the right is a regulator of gluconeogenesis. So first we're going to look over here. If you see a minus sign, that means it's an inhibitor. Now this makes sense if you look at what's going on here. This is the first step of glycolysis, where glucose is converted to glucose 6-phosphate by hexokinase. And you'll notice that over here it's showing that glucose 6-phosphate is an inhibitor of glycolysis, which makes sense because, like we discussed previously, it's a negative feedback mechanism. If you have a lot of this product of glycolysis, this glycolysis step, well, then you don't need to do more glycolysis, right? Because you already got the glucose 6-phosphate. Why are you going to waste energy and resources to make more? So that's why this product step of glycolysis is an inhibitor. Then we're going to look over here and we see a lot more inhibitors and activators. When you see a plus sign like these two over here, those are activators. When you see a minus sign, like we said before, those are inhibitors. Anything on this side is activating or inhibiting gluconeogenesis. Anything on this side is activating or inhibiting glycolysis. Now, just like I was saying in the previous slide, how products tend to be inhibitors because, well, if you have a product, you don't need to make more of that product. Well, the opposite is also true. The substrates can be activators because if you have a lot of this substrate, for instance, fructose 6-phosphate or fructose 2,6-bisphosphate, well, then you're going to want to make more. You're going to want to break that down since you have so much of it. So for instance, over here, you see that fructose 1,6-bisphosphate can be an activator to pyruvate kinase and this step over here because if you have more of that substrate, you're going to want to break it down. You're going to want to do more of the reactions that come after it. Okay? So this kind of shows you some of the activators and the inhibitors. Now, as we've mentioned previously, glycolysis is something that can be done to gain energy. You get 2 ATP out of glycolysis, and the pyruvate product can then go toward other reactions that help get even more energy, especially if it goes aerobic into full cellular respiration. So I want you to circle star highlight the fact that you keep seeing ATP as an inhibitor. See ATP again as an inhibitor to the glycolysis steps over here. And we've mentioned that before, but I just wanted to emphasize it again. Okay, so now I want to go over hormones that can regulate glycogen synthesis and breakdown. And the 3 hormones that we're going to focus on are really important for not just this class, but for a lot of the other classes you'll take, and even for graduate studies that a lot of you may end up going into. The three hormones are insulin, glucagon, and epinephrine. And so on your slide, you have the more complicated views that we're going to take a look at in a minute. But I wanted to go back to this figure from previous slide earlier in today's lecture, because I think it does a good job of simplifying the idea of what these three hormones are doing. And so over here and over here, you see insulin, and then here you see the other two hormones I just mentioned, which are glucagon and epinephrine. Now, when you look at this simplified view, it helps you see that in the case of insulin, insulin is secreted by the beta cells in your pancreas in what's called the islets of Langerhans. So you don't have to remember the islets part, just focus on insulin being secreted by beta cells in your pancreas, and that secretion is in response to when you have an increased blood sugar level. Okay, so if you have a high blood glucose, you'll have increased secretion of insulin. Now, if you have a whole lot of glucose in your blood, think logically, well, what are you going to want to do? If you just have massive amounts of glucose in your blood, that's going to be a problem. So if blood glucose is high, you're going to want to store that. Store that. And what's the stored form of glucose? Well, glycogen. So what insulin will do is when your blood sugar is high, you'll have an increase in insulin secretion, and that insulin will then trigger a cascade that eventually leads to an increase or activation of glycogen synthesis. So now that will allow that high blood glucose to get put into the stored form. Instead of glucose, you'll have glycogen. Now, the opposite is true if instead your blood sugar drops. And well, now, if your blood sugar drops, what do you need? You need some glucose available, right? So what your body will want to do is break down the glycogen now, which is this side of the pathway. That's controlled by glycogen phosphorylase. So you'll notice that the glucagon and epinephrine act to oppose the insulin pathway. So glucagon and epinephrine now, when the blood sugar is low, will activate the breakdown of glycogen, right? The breakdown of glycogen to get an increase in the blood sugar, okay? Now, when you think of that, when you think of insulin increasing glycogen synthesis and glucagon and epinephrine increasing glycogen breakdown, that means that they'll then also oppose the opposite, meaning glucagon and epinephrine you see are blocking glycogen synthesis, because instead they're for when you want glycogen breakdown, and you notice that insulin will block glycogen breakdown. Okay, that's what the minus on each of these is showing, because we mentioned earlier you don't want synthesis and breakdown happening at the same time. So if you're the hormone insulin that promotes the synthesis of glycogen, you will also want to block the breakdown of glycogen, because you don't want synthesis and breakdown happening at the same time. Okay, so now we'll just take a quick look at the more complicated figures from the textbooks. So this one, excuse me, this one is showing the exact cascade pathway that will occur to activate the glycogen synthase and the enzymes responsible for converting glucose into glycogen in the nice stored form. And then this side, this figure is to show glucagon and epinephrine, the pathway that they will trigger in order to then break down glycogen into glucose. Okay, so that kind of shows you the two sides of the story and what happens. And like I said, a lot of this is kind of thinking of the logic of, well, okay, what does the body have at that time? Does it have a lot of glucose and need to get rid of it? Does it have low glucose and need to get more? Once you think of that, you'll then be able to think of, okay, what gets me glucose versus what stores it. And so kind of keep that in your mind to figure out the different roles of these hormones. Now, here's a recap of the main points that I just made in those slides about the 3 hormones and exactly when they will be activating certain pathways, when they'll be blocking them, and what exactly they're doing. So please make sure to circle, star, highlight this recap because it has a lot of important information that I could ask you about. Okay, so while we're on the topic of regulators and controlling the pathways, I just want to give you a copy of this table from your textbook, which shows 4 types of control. These are all forms of controlling pathways that we've basically been talking about in each of the previous lectures as well. Things like allosteric control, which is when you, for instance, have inhibitors or activators that can bind to the enzyme in a pathway and change its shape or increase or decrease its efficiency. You have covalent modifications, which are also going to be either inhibiting or activating an enzyme, and a lot of times can be irreversible. You have substrate cycles, and so that's kind of along the ideas of the negative feedback that we keep mentioning of how products can block reactions and how having more or less of a substrate can affect whether or not that reaction can occur. And then you have genetic control, and those of you who have had me for other classes know I love anything related to genetics, but you can basically have genes and promoters, controlling how much of an enzyme you have, basically based on gene expression control. Okay, before we leave glycolysis and move on to the citric acid cycle, I just want to mention one more pathway, and there's going to be a recap slide on this slide in a minute. The pathway I want to mention is the pentose phosphate pathway, which is represented here. And in the pentose phosphate pathway, it's basically an alternative to glycolysis, and it differs from glycolysis in multiple ways. So as you can see here, we have glycolysis labeled out over here, whereas the pentose phosphate pathway, a big difference is that, as the name suggests, instead of the goal being glycolysis and making ATP, energy for the cell, the main goal for the pentose phosphate pathway is to make a pentose such as ribose or its derivative deoxyribose. And that's really important because that means that this pathway, the pentose phosphate pathway, goes toward building nucleotides, okay? Because as you know, DNA, RNA, they rely on ribose or the derivative deoxyribose. Another important difference is that a big part of the pentose phosphate pathway is the production of NADPH, so as opposed to NADH. And what NADPH is nicotinamide adenine dinucleic phosphate. And so it basically has an extra phosphate group compared to the NADH that you kept hearing mentioned in the glycolysis pathway talks. Okay, so the big difference is emphasized here on the recap slide. And so you can pause and jot down these notes. And then we're going to move on to the citric acid cycle now. Okay, so now we're going to head into the citric acid cycle, and you may have seen this cycle called the Krebs cycle or the TCA cycle, tricarboxylic acid cycle. And I'm going to have a recap slide next so you can see the spelling of all of these terms. Now it's called sometimes the tricarboxylic acid cycle because some of the molecules involved are acids with 3 carboxyl groups, so tricarboxylic. Okay. Now ultimately, the citric acid cycle or Krebs cycle or TCA cycle, whatever you want to call it, is significant because it plays a role in aerobic metabolism. And if you picture the idea of, glycolysis, prep reaction, citric acid cycle, electron transport chain, in terms of, when you learn these things in intro level bio, you'll notice that the aerobic pathways end up producing a whole lot more ATP for a cell. So for instance, glycolysis can produce 2 ATP, but if you go the whole route of cellular respiration with the citric acid cycle and then the electron transport chain, which you see in this figure here, you can end up with 36 or 38, anywhere from 32 to 38 ATP produced, which is a lot more than a measly little 2 ATP. Now, this cycle is also important because it's linked to metabolism, catabolism, anabolic reactions and all. And so when we kind of think about how it fits into metabolism or catabolism, this little figure here shows that a lot of the products of catabolism, so glucose catabolism, fatty acid, amino acid catabolism, fancy way of saying the breakdown of these biomolecules then goes into the citric acid cycle. And once they're in the form of acetyl-CoA going into the citric acid cycle, once you've had the citric acid cycle, you have these electrons, these carriers of NADH and FADH2 that could then go into the electron transport chain. And like I just mentioned, the electron transport chain and the oxidative phosphorylation pathway, that's where a cell could get a whole lot of ATP energy. It could get like 32 ATP from this single initial glucose molecule. Okay, so the idea of catabolic events being linked, you can kind of see that you need the breakdown of some substrates to then end up carrying into the citric acid cycle to then lead to more energy for us. Now, even though I keep saying catabolism like a million times right now, sorry about that, or the breakdown of these molecules, the citric acid cycle is actually amphibolic, and amphibolic is a fancy way of saying it plays a role in both catabolism and anabolic reactions or anabolic pathways, because some of the molecules that are going to be included in this cycle, they end up being starting points of biosynthetic pathways, meaning anabolic pathways as well, so not just the breakdown. As promised, here's a recap of that information from the past slide so that you can pause and jot down your notes. Now, before delving into the exact steps of the citric acid cycle, it's important to do like we did with the previous cycles and kind of question and visualize where these events are going to be occurring. So the citric acid cycle, you notice I wrote, takes place in the mitochondria. A lot of the steps within the citric acid cycle you'll specifically see taking place in the matrix of the mitochondria, but there are a couple or at least one that'll be in the inner membrane area. So inner membrane highlighted over here. And we'll go through that in more detail in the upcoming slides, but basically the step in which where the electron acceptor has to be FAD, it has to occur inside the mitochondrial membrane because the particular enzyme is located there. Which brings us to the idea of how this differs to glycolysis, because you remember in glycolysis and the gluconeogenesis pathway as well, we were looking at the cytosol. And remember, sometimes certain enzymes, like the enzymes needed in terms of converting pyruvate to oxaloacetate, they are located in a particular place. So those enzymes were in the mitochondria, whereas we needed the oxaloacetate to be out in the cytosol. So events like glycolysis, they occur out in the cytosol. And that's why in intro level bio, when we go through things like cellular respiration, we always show glycolysis kind of separate and on its own because it's out in the cytosol and it's anaerobic, whereas the rest of the steps of the prep cycle, the citric acid cycle, and electron transport, they all occur within the mitochondria and are aerobic. Okay, so here we have an overview of all of the reactions in the citric acid cycle, or the TCA cycle, or Krebs cycle, whichever name you're most comfortable with. But just make sure you're aware that they are all names for the same cycle. Be able to recognize all three. Circle star, highlight that fact. And you'll notice when we're heading into the citric acid cycle, we're basically taking over from where we left off previously in terms of glycolysis. You at the end of glycolysis have pyruvate, and we had mentioned that pyruvate is a very powerful molecule. It has a lot of options when it comes to its future fate. And one of them is to head on into the citric acid cycle, first as, converting to the form of acetyl-CoA, and then heading through. And you'll notice, just like with each of our other cycles that we talked about, each reaction within the cycle has its own particular enzyme. And so that'll be important when we go through controls. And overall, you see 8 different steps with 8 different enzymes that'll be a part of this whole process. And you see some familiar players that we've mentioned before, things like malate and oxaloacetate are coming back again now. So now pyruvate, like we keep saying, is a very significant molecule because it's got a lot of different fates, and it can come from a lot of different places as well, one of which that we've mentioned is, of course, glycolysis. It moves from the cytosol into the mitochondria. We saw that previously as well in those slides where eventually it became oxaloacetate and malate and that whole conversion there. But now we're going to focus on it being converted to acetyl-CoA within to get it into the citric acid cycle. And you'll see this guy will keep coming up as very significant. The pyruvate dehydrogenase complex, which will be responsible for this conversion. It's a complex of 5 different enzymes. You're not going to have to know the exact enzymes, which, if we listed them out, would be pyruvate dehydrogenase itself, PDH, as well as dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase, pyruvate dehydrogenase kinase, and pyruvate dehydrogenase phosphatase. And the reason why I list them out is so that even though you don't have to know them or recognize those particular ones, I want you to be aware that out of the 5, 3 are for the actual conversion that you're seeing here of pyruvate to acetyl-CoA. But then those last 2 that I mentioned, the kinase and the phosphatase, are actually responsible for regulating and for controlling the pyruvate dehydrogenase itself, because remember, that was the very first enzyme out of the 5 that we did mention. Okay, and regulatory molecules are always important for us to have, these activators, inhibitors, enzymes to kind of control whether or not the system is running. Okay, now the fact that this conversion is happening from a complex of 5 enzymes instead of one single enzyme is important. It gives it a strong advantage. Instead of having the linear aspect of, let's say, 5 completely separate single enzymes, now you end up with this complex where there's a compact arrangement of the enzymes within the complex. And so now you have reactants and enzymes closer together, which will increase the efficiency and make things happen much faster. And you also have regulatory controls being more efficient because, again, when everything is nice and compact and within a nice single um, multi-enzyme structure, you end up having more areas to regulate, basically, more areas that you can activate, more areas that you can inhibit. Okay, so I know we've already seen an overview of the citric acid cycle and all of the reactions in their actual chemical structure formulas, but I like this table because it nicely maps out all 8 steps. Remember that I said that there are 8 steps in the citric acid cycle, each with their own enzyme. So circle star highlight the fact that there are 8 steps and 8 enzymes in the citric acid cycle. I want you to write down some significant points. So I'm not going to make you memorize every single step, but I do want you to make note that FADH2 and NADH are made in the citric acid cycle because FADH2 and NADH are going to be very important in a couple of slides. And I want you to kind of recognize the enzymes that are involved. Okay, so when you see something like citrate synthase, that is part of the citric acid cycle. Okay, but like I said, you're never going to have to write out or memorize each of the steps that you see here. Now in biochemistry, it's important whenever we look at any kind of cycles or reactions to ask ourselves about the energy or the energetics and the kinetics of these particular reactions and these cycles. So when we talk about the citric acid cycle, before we got into the 8 steps of the citric acid cycle, first we saw that you need to have pyruvate converted to acetyl-CoA. So that initial reaction before the citric acid cycle, when you look at the energetics of it over here, the free energy, you notice it is negative. And earlier in a previous lesson, we mentioned when you see a negative for the free energy of a reaction, that tells you that it is an exergonic reaction. So this pyruvate to acetyl-CoA reaction, because it's negative here, that is exergonic. And then when we look down and we look at the overall reaction, if you subtract or if you get rid of that negative 33.4 from this negative 77.7 to just look at the 8 steps of the citric acid cycle, you will still end up with a number that's in the negative. Okay? So for instance, over here, if you're looking at -8 is this first reaction, the overall of all 9 of these steps is -18.6. So the citric acid steps are just -10.6. So that is still negative. Okay, and here you have -44.3. So that's still negative for the citric acid cycle steps. Okay, so the answer to both of these questions is exergonic because the free energy of these overall cycles and these reactions are negative. So just like a lot of the other slides and pathways that we've talked about, the citric acid cycle is controlled in a way that's really logical if you think about what a cell needs at that moment and what the pathway is actually doing. Now, in this overall pathway, you'll notice that just like we've been talking about, a lot of times a product of a reaction, so for instance, in this case, citrate is a product over here, that will then be an inhibitor to the previous reactions, okay? So as you can see here, acetyl-CoA to citrate, citrate is listed as one of the inhibitors to this pathway because once you have enough of a product, you don't need to waste energy and resources making more. And you'll notice that because the citric acid cycle is ultimately part of a pathway to make ATP an energy for a cell, you'll see throughout the cycle that ATP in each of the steps is a big inhibitor for the citric acid cycle. If the cell has a lot of ATP, it doesn't need to go through this huge cycle to make more. Why would I waste my energy and resources making what I already have? Okay, so you'll notice that ATP is a big inhibitor for that reason, and NADH is also a big inhibitor that comes up in each of the steps. And then the other inhibitors that you see, like I mentioned before, are mainly the products of certain steps. Now, when we look at this, you can also think about the cell itself in terms of its metabolic states. So there's resting metabolic state when, a cell is chill, it's not really using much energy, it doesn't need much energy, versus when it's highly active and it needs to get things done, right? So when you look at these, it goes along with our logic. If a cell is in the resting state, it's chill, it's not using much energy, doesn't need much energy, it already has plenty of ATP and NADH available. So in this resting metabolic state, you would be inhibiting the citric acid cycle. Okay, because we said whenever you have a lot of ATP, so a lot of ATP or a lot of NADH, you are inhibiting the cycle. You don't need to waste your energy making more. The opposite of that is a highly active metabolic state. If you're active, you need energy, you need more ATP. So in that scenario, you'll consider the ATP and the NADH too low, so then you will activate the citric acid cycle to make more. This is a recap of those concepts, and make sure you're very comfortable with what conditions would inhibit the citric acid cycle, what conditions would activate it, okay? Because those are easily made into exam questions. Now, because you can never have enough cycles, there's another cycle to mention. There's the glyoxylate cycle. Basically, that's a cycle in which some organisms can make carbohydrates from acetyl-CoA. So they convert the acetyl-CoA into carbohydrates. Plants and some bacteria can do that. Animals cannot do that. So animals can convert carbohydrates to fats, but they cannot convert fats to carbohydrates. And acetyl-CoA is, that pathway is considered the catabolic reaction of fatty acids. So animals cannot do this. So circle star highlight the fact that we can't because we are animals. We cannot do the glyoxylate cycle that only plants and some bacteria will. And as you can see, basically there are two main reactions to be aware of in this cycle, and so there are two enzymes involved in this cycle. You have isocitrate lyase for the conversion shown up here to produce succinate and glyoxylate, and then you have malate synthase to produce our little buddy that we have seen before, malate. We saw that in terms of the oxaloacetate reaction that we looked at earlier. Now, this is important or valuable that it produces malate over here, because then that malate can go on to be converted to the oxaloacetate that we've mentioned previously. And the reason we mentioned that previously is that that molecule can then go into gluconeogenesis, which allows the cell to then produce glucose. Okay, so it's a, a nice little way for them to be able to get some glucose. Now, the last few slides of this lecture are just to quickly show you how everything in that cell is basically connected. We keep mentioning catabolic reactions and anabolism in terms of metabolism, and so this shows you how catabolism is connected to the citric acid cycle. You'll notice that both events take place in two different places. So catabolism will occur out in the cytosol, whereas this little pink kidney bean shaped structure here is the mitochondria. So all of these reactions that you see out here, these are to show you catabolism occurring in the cytosol. You'll notice that the catabolism of carbohydrates, of lipids, so I forgot where they put lipids over here, and then of various amino acids. So you have the various amino acids all over here. And so with that, you get to see all of these catabolic events. Oh, there's the lipids that I was looking for earlier. So it shows the catabolism of lipids, carbohydrates, and then all different amino acids. So all of these guys around the edges here are amino acids getting broken down, and then their products can end up crossing the mitochondrial membrane, which you see these big purple arrows showing the products of catabolism crossing the mitochondrial membrane and serving as intermediates or substrates and reactants within the citric acid cycle. Okay, so for instance, lipid catabolism feeding the acetyl-CoA reserves to head into the citric acid cycle. And same thing with certain amino acids, also feeding more acetyl-CoA. The breakdown of carbohydrates feeding pyruvate into the cycle. Okay, so you see catabolism is a big feeding source for the citric acid cycle. Now, just like catabolism is an important way to get what we need in terms of the citric acid cycle, so is anabolism or anabolic reactions, which is building things up. You'll see here a term that I want you to know, which is anaplerotic reactions. That's basically any reaction that will yield an important intermediate of metabolic reactions. Okay, so basically, if something is producing, for instance, the intermediate malate here, then that would be a reaction that's considered anaplerotic. And those reactions are basically to ensure that you always have the right supply of these important metabolites. And if you think about it, you know, the citric acid cycle also plays a big role in anabolism by providing starting material for the biosynthesis of many other biomolecules. So just like before we saw all of the arrows pointing inward, we're also able to see a lot of arrows pointing outward as the metabolites and the various intermediates from the citric acid cycle can be used to build up things like fatty acids and lipids or carbohydrates or various amino acids. Because like we always say, you know, in biology, you have both, you know, opposite goals occurring within a cell. You know, for every reaction, there's an opposite and equal counter reaction. Okay, so that's why it's important to really connect the big picture of all of these events. The last thing that I want to mention for today's lecture is the link to oxygen, because, you know, sometimes students get a little confused by how, especially, you know, in the earlier introductory undergrad courses, you know, we emphasize, okay, the 4 steps with cellular respiration, glycolysis is outside the mitochondria and the cytosol, it's anaerobic, whereas the other 3 steps to that cellular respiration, the prep reaction, the citric acid cycle, electron transport, they all are aerobic and within the mitochondria. And yet today, when we went over the citric acid cycle, we didn't see oxygen. So why do we say aerobic if we're not seeing oxygen? But basically, the reason why citric acid cycle is considered aerobic is that it's closely tied to the electron transport and oxidative phosphorylation steps of this whole big pathway, which eventually do lead to oxygen. And so the citric acid cycle we mentioned produces NADH and FADH2. And those serve as the electron carriers for the heavily aerobic electron transport chain and oxidative phosphorylation. That's the next step. Okay, and so they're basically handing off electrons to oxygen in those next steps that we're going to talk about. And so that's why the citric acid cycle is considered to be aerobic. So even though you're not directly seeing molecular oxygen in the reactions we talked about, you're seeing the citric acid cycle as part of a big aerobic process because its NADH and FADH2 are then electron carriers for the reactions that do have molecular oxygen in them. And that is it for today. Thank you. And as always, if you have any questions, contact me in the Remind app anytime, day or night. Always say I have no life. All right. Thank you and have a great day.